Our next speaker, many of you know, I hardly feel like I have to make an introduction, but I will. Dr. Doug Wallace, he's the director of the Center for Mitochondrial and Epigenomic Medicine. He's the Michael and Charles Barnett Chair for Pediatric Mitochondrial Medicine and Metabolic Disease. His training is he had a Bachelor's of Science degree from Cornell, a master's degree from Yale, a Ph.D. in microbiology and human genetics from Yale, and then an NIH fellowship. He has been interested in mitochondria and the role of mitochondria in diseases for 45 years. He has numerous awards. He's a member of the National Academy of Science. He has membership to the American Academy of Arts and Sciences, membership of the Institute of Medicine, the Gruber Prize for Genetics, and then in 2017 he got 2 prizes, the Benjamin Franklin Life Science Medal and the Dr. Paul Janssen Award for Biomedical Research. And today he's going to tie a lot of this together and talk to us. The title of his talk is Mitochondria as Chi, a Mitochondrial Bioenergetic Etiology of Disease, East Meets West.
Well, thank you very much for the introduction, and I hopefully not disappoint you. But since yesterday I had a chance to speak also, I've decided to come back to my roots and talk about ophthalmology. After all, it is an ophthalmology meeting. And I'm going to start with a disease that we have been interested in now for actually 40 years, although it's the 30th anniversary of finding the cause of this disease, and that's Leber's hereditary optic neuropathy. And this disease, as I'm sure many of you know, is really a stealth disease. You don't know, you as the future patient does not know that he primarily or she is at risk for this disease. And then it has a rather spectacular onset of sudden onset of central scotoma in one eye, and then within a year, usually often much less, a central scotoma in another, with very rare instance of regression back to normal. So this is pretty devastating. It occurs in midlife, often when people are young in their career or in their marriages, and really radically alters the quality of life. This disease is fascinating in many different ways, though its actual incidence as far as the practicing ophthalmologist is concerned is not all that frequent, about 1 in 30,000. In fact, the number of people that actually carry the mitochondrial DNA risk factor for that is 1 in 10,000. So it actually turns out to be one of the most common mitochondrial DNA disease risk factors. It has a number of features. One of the interesting things about it is if you use OCT, prior to the onset of the acute event, you'll see swelling of the optic disc, and then later you see the optic atrophy and the loss of that swelling. And so there's a lot of interest in how the pathophysiology relates to this pre-onset swelling phenomena, although I must say we don't really understand that very well right now. What was most interesting to me in the 70s when I started getting interested in this disease is the pedigree. The pedigrees, as Dr. Malik said, are often important to look at. And what was found, this was a pedigree. Actually, this is our type pedigree that worked on in the late 70s. Where you can see that these people that are filled in squares have the sudden onset optic atrophy. This individual is related through this female to this female, and these 2 affected individuals, through this female and these affected individuals, this female and all these affected individuals, and this female here to this affected individual. Now, this disease is rather unique because there's a 4 to 5 to 1 male to female bias. So in the literature, this disease was attributed to X-linked inheritance because it's transmitted through the female lineage and the males are preferentially affected, and that was classic Mendelian presentation. The only oddity is that about 20% of the people that actually go blind are females, and by X-linked inheritance, they should have been heterozygous X-linked, and they should not have gone blind. So that was a hint to me that this was not exactly the right decision. And so we then had shown at that time that this piece of DNA, the mitochondrial DNA, is inherited from the mother to all of her children and her daughters to their children. So you might expect a mitochondrial disease to affect all the maternal lineages. But we had also been studying the fact that each cell has 5,000 mitochondrial DNA copies, and you can have different percentages of mutant and normal. And so then you get a graded phenotypic expression, and then you could have people who carried the genetic trait but had a lower percentage of mutant and did not, in fact, express the phenotype. So at the time, in the 70s, I hypothesized that that was what was going on. Actually, I was only partially right. It is a mitochondrial DNA mutation, but the idea that it was heteroplasmy that caused this effect was absolutely wrong. But that's okay, because it still didn't fail to find the mutation. So the mitochondrial DNA is a circular piece of DNA of our genome, and it's very important in generating energy, as Dr. Malik had mentioned. So the question that has often been asked is, why could a systemic bioenergetic defect, like the one that's caused by the mitochondrial mutation of Leber's, give you a specific phenotype, in this case, delayed onset acute optic atrophy? And Valerio Corelli and Alfredo Sadoun have been quite interested in this for quite a while, and they've come up with this hypothesis based on their anatomical analysis of patients. So this would be what you would see if you stain for myelin basic protein of the optic disc of a normal individual and of a patient with Leber's, and you can see that the loss of the high energy central optic nerve retinal ganglia cells. And then this would be the myelin around a normal optic nerve, and you can see you get demyelination around the optic nerve of these affected individuals. And here you have only a few mitochondria, and then you see the optic nerve swell with the proliferation of these abnormal mitochondria. Now, whether the mitochondrial proliferation gives the swelling or not is still unclear. So the question then is, well, why would then the retinal ganglia cells be so prone to this sudden onset death? And their idea is that it relates to the anatomy of the eye, and that is when God gave us eyes, he made a mistake and made them upside down. So then the optic nerve fibers are on the top, and the light has to go through the optic nerve fibers. So that's a big problem, because if you myelinate them, then you have a lot of lipid, and that's going to diffract the light, and then you're not going to be able to see. So what happened evolutionarily is we got rid of the myelin on the fibers that are across the surface of the retina. That has a problem, though, because the beauty of myelin is it's a very efficient way of conducting electrical signals along nerves. So you put myelin as an insulator, and then you pack your mitochondria at the nodes, and then every time you fire your neuron, then you use sort of an electrostatic signal to fire each of the nodes of Ramnion, and thus you save a lot of energy and increase the transmission rate. But up here, if you're going to get rid of the myelin, you have a problem, because now you need a lot more energy, because now you have to have the action potential move across the entire surface. So this area then becomes packed with mitochondria. So the idea was that the mitochondrial dysfunction then what would preferentially affect this system where you had the highest energetic demand. I think that's a good hypothesis. I don't know that we've been able to actually determine that in any physical way. So anyway, why did all of this develop? Well, it developed because when our cells got formed, our eukaryotic cells, that was an invention of evolution by the marriage of two totally different life forms, an archaeobacteria and an oxidative eubacteria. And the archaeobacteria ultimately gave rise to the nuclear cytosol, and the oxidative bacteria gave rise to bacteria inside your cells called mitochondria. And these mitochondria then proliferate in the cell, and they generate in the order of 90% of all the energy that we use in our body. Now, because it's a bacterium, it has its own DNA, which is replicated in the mitochondria, transcribed into transfer RNAs and ribosomal RNAs, and 13 polypeptides that are read on mitochondrial ribosomes. And these ribosomes are bacteria-like in that they are initiated by N-formyl methionine, and they are sensitive to chloramphenicol and aminoglycosides and a lot of classic bacterial inhibitors. There are 13 polypeptides in the mitochondrial DNA, and those are the key electron and proton wires of energy production. Hence, we refer to it in our lab as the wiring diagram of the power plant. It turns out that these 4 enzymes, which we're not very good at clever names, but this is complex 1, 3, 4, and 5, and we also can't count because we left out 2. But anyway, the point is that these are made of multiple polypeptides, some coded in the nuclear DNA and others in the mitochondria. So this enzyme, complex 1, is the most complicated enzyme known to, I guess, biology. It's 45 polypeptides, and 7 are coded in the mitochondrial DNA, the rest in the nucleus. This enzyme is 11 polypeptides, 1 in the mitochondrial DNA, the rest in the nucleus, 13 polypeptides for complex 4, 10 in the nucleus, 3 in the mitochondria, and complex 5, 15 in the nuclear, and 15 in the nucleus and 2 in the mitochondria. So we have this very strange situation of 2 different genomes, this genome having about 1 to 2,000 genes that go to assemble the mitochondria, plus the mitochondrial DNA that actually codes for the wiring diagram. This is a classic concept of how we make energy, and every medical student I've ever taught hated this, so I'm sorry, you're just going to have to go to sleep. But basically, we burn in our mitochondria hydrogen that plants put onto carbohydrates using the energy of sunlight, and we oxidize that hydrogen with the oxygen that we breathe to give water, and the energy that's used is made to create a capacitor, and it's all very easy to understand. The glucose goes through glycolysis to pyruvate. Pyruvate can get reduced to lactate or get an amino group to give you alanine, but then it goes through pyruvate dehydrogenase to drive the tricarboxylic acid cycle. That strips the hydrogens off the hydrocarbons, puts them on the carrier NAD, and then we burn these 2 hydrogens through this electron transport chain, complex 1, CoQ, complex 3, cytochrome C, complex 4. Now, then to oxygen to water. Now, the purpose of this is then to use the energy or trap the energy by making an electrochemical gradient that's positive and acid on the outside and negative and alkaline on the inside. And that potential energy then is used for everything that you do, including your retina, which is probably the most oxidative tissue in your body. So this electrochemical gradient then can be used by the ATP synthase to condense ADP and phosphate to make ATP, and then the ATP goes out through the translocator where it's used to do work, and it comes back in to be rephosphorylated. So you're coupling the electrochemical energy of oxidation with phosphorylation, hence oxidative phosphorylation. Now, one of the interesting things about this is that this oxidation is often incomplete. Just like any combustion in any furnace, you're going to get some smoke, and this is going to be very important for us to understand labors and we think glaucoma, and that electrons from complex 1, CoQ, and complex 3 can be donated directly to O2 to give you superoxide anion, which is a potent oxidizing agent. Then superoxide anion can be condensed, 2 of these, to give hydrogen peroxide, which is more stable. But there's a lot of transition metals that are reduced in the mitochondria, and they can then add another electron to give you hydroxyl radical, and these are the potent oxidizing agents. And those are very important reasons for why, as we heard earlier, why you might take vitamin E, vitamin C, and if you don't worry about lung cancer, beta carotene. Trying to remember what you said.
I want to remember that.
Yeah. And so this enzyme, hydrogen peroxide can be detoxified by glutathione peroxidase. The mitochondria also picks up calcium, and calcium is a major regulator. One of the things I asked Dr. Malik yesterday is, what is a calcified plaque made of? Obviously, calcium is one of the things, but there's probably a lot of other interesting things going on to get this kind of opaque plaque. We also have then a self-destruct system. Nobody knows what it is really made of, but it measures the membrane potential, the ATP, the calcium level, the oxidative stress, and when they get out of whack enough, it opens a channel, short circuits the membrane potential, backs and back, form a megachannel, release proapoptotic proteins, and they degrade the cell from inside out. So basically, from the point of view of thinking about labors, we need to understand that the mitochondria makes energy, yes, but more importantly, it regulates redox balance, reactive oxygen species, regulates calcium, regulates apoptosis, and also all the intermediates of the mitochondrial, not all of them, but many of the intermediates of the TCA cycle are used to regulate nuclear gene expression. So this is the mitochondrial DNA. As I said, it's maternally inherited. There are thousands of copies, so you can get a mixed population we call heteroplasmy. And when the cells divide, it's statistically a matter of chance where the mutant and normal will go. So you can get tissues with different percentages of mutant and normal. And the more mutant you have, the less energy. And hypothetically, as energy falls below the minimum for that tissue, then you'll get a phenotype. So we call that the bioenergetic threshold. But this is the important thing for today's discussion. We have the mitochondrial DNA, for which you have thousands of copies in each cell. You have ribosomal RNAs, transfer RNAs, and ND1, 2, 3, 4, L4, 5, and 6 for complex 1, cytochrome B for complex 3, CO1, 2, and 3 for complex 4, and ATPase 6 and 8 for complex 5. So getting back to labors then, the most common cause in Western Europe of labors, and also in Asia, by the way, is a nucleotide position at nucleotide position 11778, a G to A transition. But you can have labors for lots of other reasons. You can have a mutation in the ND6 gene at 14484, T to G, or you can have it in the ND6 gene at 14459, G to A. These are relatively mild, and they give you the blindness when they're pure mutant, homoplasmic. This one is more severe, and it gives you blindness when it's heteroplasmic, when there's a mixture of the two of mutant and normal. When it becomes homoplasmic, pure mutant, it gives generalized dystonia. Okay, well, there are lots of other mutations, but the other point to be made is there are also these ancient polymorphisms, like this variant identifies all mitochondrial DNAs from Africa. We call that macrohaplogroup L. This variant is found in half of Europeans. We call that H. Variant A, B, C, and D arose in Central Asia. They crossed the Bering Land Bridge, gave rise to Native Americans. And these variants then tag lineages that then predispose to different kinds of diseases. And finally, we accumulate somatic mutations with age. So why do we have age-related disease? Sorry for all the background from those who were yesterday, but we need to get these points made. The brain is 2% of your body weight, uses 20% of all your mitochondrial energy. Therefore, the first tissue to be affected is the brain, and even more so, the retina. And then followed by the heart, muscle, renal, and endocrine system. There are systems for energy generation to make ATP or to generate heat, or regulate blood glucose in the liver, and of course, sensing the levels of carbohydrates in your environment using the pancreas alpha and beta cells. So basically, our working hypothesis is that a large array of different kinds of common complex diseases may have as their underlying etiology bioenergetic defect rather than anatomical problems. And this could be then associated with changes in the nuclear DNA or changes in the expression of the DNA through the epigenome, variation in the mitochondrial DNA, either ancient adaptive polymorphisms or recent deleterious mutations. But as Dr. Malik said, as soon as you're beginning to think energetically, then your diet becomes critically important, or whether you exercise or not, or whether you're under stress. All of these things affect mitochondrial function. Particularly bad is if you smoke, because if you smoke, then there are lots of toxins that are going to inhibit oxidative phosphorylation. If you inhibit oxidative phosphorylation, that is mitochondrial energy production, you're going to perturb mitochondrial DNA replication, and that's going to cause the mitochondrial DNAs, as they replicate, to accumulate damage. And that age-related accumulate damage, then, we believe is aging, and also is why many of these diseases have a delayed onset and progressive course. Because why would you inherit something like a nuclear gene mutation, but then wait until you're 60 to have a phenotype? There has to be some other clock mechanism, and we think it's the accumulation of this mitochondrial DNA damage. So it's going to affect then your central nervous system, your heart, your muscle, and renal systems, because those are all your high-energy tissues. If you impair energetics, then your substrates are going to build up, and those are going to be glucose, triglycerides, cholesterol, and that's going to give you metabolic syndrome. Your mitochondria is the most common bacterium in your body, and if you release that in your bloodstream, you're going to have all the bacterial inflammation effects, and that's why all these diseases have inflammation. And finally, cancer is all about having enough energy to grow. So that's a basic working hypothesis. Okay, well, this is one of the early pedigrees we worked up. This woman presented, this gentleman presented with us with optic atrophy and spicular retinitis pigmentosa as a major presentation. This is this gentleman here. When we did MRI, you can see that the cerebellum has been reduced to about a third of the volume. The brainstem is about half its volume. That's olivopontocerebellar atrophy. This person, of course, was devastated and in a nursing home. But actually, that was not the person that we actually was presented. This mother presented with these two kids in tow at our clinic at Emory. This boy was in her arms, pretty unresponsive, and that is this boy. And this little boy was wandering around the hall looking like this. So he had cerebellar ataxia. So he just walked down the hall. We made the diagnosis. Nancy Newman, my long-term collaborator, then did a complete retinal workup, and all of these people had retinitis pigmentosa. So we ultimately found out that had a mutation in the mitochondrial ATPase6 gene at 8993, a T to G transition, and that changes the leucine at codon 156 to arginine. And what was interesting is that all these people with just retinitis pigmentosa through the maternal lineage, not realizing or even thinking that they had a problem, they were about 75% heteroplasmic. That is, they had 75% of the ATP synthase mutant and 25% normal. And yet they were essentially phenotypically normal. Of course, we didn't ask them to run upstairs or anything. But what was interesting, pardon?
They had RP or not visible?
They had salt and pepper RP. Okay. That was what Nancy's diagnosis was. But this mother gave a family history that this boy had died in the same way as this non-responsive boy in her arms, which is why she came to our clinic. And on examination, this boy has central basal gangliar degeneration, classic Lee syndrome. And unfortunately at the time, and even now, the prognosis is dire. But this boy had then, of course, cerebellar ataxia. And these people, when we looked at the percentage of their heteroplasmy, they were 90 to 100% mutant. And this person then ultimately was in between them, about 80 to 90% mutant. So a few tens of percent of heteroplasmy was the difference between just having retinitis pigmentosa and lethal pediatric disease. And so that shows how sensitive this energetic system is to how sensitive the body is to subtle changes in energetics. This is an interesting mutation because it changes this leucine to an arginine. Leucine is is neutral, and the proton uses this glutamate to get from the outside to the inside. So it goes to the glutamate and then into the matrix of the ATP synthase. But if you mutate that leucine to an arginine, now you create a salt bridge of positive charge with a negative charge and block the protons, and the ATP synthase becomes non-functional. So it's really a beautiful story about how now we understand the pathophysiology of this disease quite well. But this was the pedigree that we first worked up after I started thinking that Leber's might be a mitochondrial disease. This was a pedigree we did while I was at Stanford. And these people had optic atrophy, which is how they came to our attention, classic Leber's, as did this person. But there was these other odd people here. This woman had Leber's optic atrophy, but then her two children had generalized dystonia. And then here we have a woman that had dystonia with children with dystonia. Here we have a situation where we have a normal mother with optic atrophy and two children with dystonia and one with both. And then with a different husband, 3 children with optic atrophy and 1 unaffected, and on and on. So you have this very, very odd phenotype, or phenotypic progression of both optic atrophy, optic atrophy, generalized dystonia, and normal segregating along the maternal lineage. So when we first found this pedigree, I was thrilled because I now thought we had proven maternal inheritance of Leber. So I went to my ophthalmology friends and said, Eureka, we've now shown maternal inheritance of Leber's hereditary optic neuropathy. Oh my God. Optic neuropathy. And they said, they looked at the pedigree and they scratched their head and they said, that's ridiculous. These people don't have Leber's. Therefore, it's not maternally inherited. It can't be mitochondrial. And first of all, we never go to parties with those neurology people. And so then I thought, well, maybe neurologists would be more sensitive. So I went to the neurologist and said, Eureka, we have a neurological disease, generalized dystonia. They said, no, ophthalmologists are never true. They lie. And can't be a mitochondrial disease. So anyway, I was very depressed until I worked with these poor people. This woman was coming out of her wheelchair, and she's quite rigid. And this was, I think, maybe the first time bilateral basal ganglia was observed. But here was her MRI. And we called that at the time bilateral basal ganglia. Now it's called, anyway. It's now pathognomonic. Oh my God, I'm really pathognomonic of Leigh syndrome. Anyway, we ultimately found that this was due to a mutation in the ND6 gene of the mitochondrial DNA, and it changes very highly concerned alanine to a valine on codon 72. And when it was heteroplasmic, it gives you Leber's, and when it was homoplasmic, it gave you generalized dystonia. And so that's why you were getting the segregation. But interestingly enough, this is a classic Leber's mutation. It changes codon 64, relatively mild methionine to valine, but it will only give you Leber's when it's homoplasmic. And that makes sense because this is a much milder mutation, right? So that shows the quantitative effect of this. So back to our type pedigree that we were working with. Here is a disease that's homoplasmic with a 4-to-1 male to female ratio. ND4 gene mutation, 11778, G to A, changes arginine at codon 340 to histidine. So this then has many interesting aspects. It's pure mutant, but there's bias of males, and there's variable expressivity. It even got worse because, in fact, there are multiple different mutations. Now there are probably about 20 different mutations that in the mitochondrial DNA that can predispose you to Leber's, but these are 3 classical ones. This is the most severe, and if you get this mutation, you're just going to go blind. If you get this classic common one, 11778, it's more moderate, and you have a pretty good chance of going blind. And this 14484 is the mildest mutation, or one of the mildest, and has a low biochemical defect and a low penetrance. However, these will increase the penetrance if you have these background mitochondrial DNAs. This is haplogroup J, and you can see that this J is 10% of the general population here, but if you're 11778 and go blind, you're about a third J, and if you're 14484, you'll be 3/4 J, and there's a milder mutation that always is J. If you're not J, then you'll have other mutations such as this 3394 mutation in the ND4 gene. So now we have all kinds of complexity. How come there's variable penetrance? How come there's male effects? What does the background have to do with it, et cetera? So what is J? Well, we'll get to that, but let's talk about getting hybrids, taking cells from the patient with a mutant mitochondria, putting them in a cultured cell and studying the biochemistry. So this is some studies we did with the 3460, 11778, 14484 in the control. And if you look at complex 1, you can see the more severe mutation really knocks the dickens out of complex 1. But these other 2 are relatively mild. They give a relatively mild effect. And if you make these hybrids, that is, you have a uniform nucleus in the mitochondrial DNA, you can see that the same biochemical phenotype is transmitted. So 11 here and 12 here, et cetera. If you now look at the ATP production of cultured cells, this is the normal cell line, this is the 14484, this is 3460, and 11778. So now you can see that even though complex 1 is affected, there's a differential effect on ATP. So you're getting some kind of odd interactions here. This is looking at oxidative stress, and you can see that the 11778 and 3460 have much higher oxidative stress than control in all the different tests. And also, they have a more high prone to apoptosis. So this is the 11778 and 3460 death by apoptosis. So now we're beginning to see how the mitochondria is resulting in the pathophysiology. But there are all kinds of interesting aspects about why there's variable penetrance. One of the most greatest environmental risk factors that Freda Sedone and Valerio Corelli found was smoking. So smoking will increase your penetrance substantially. Also, if you're a very heavy drinker, you will increase your penetrance. But also, if you take certain kinds of antibiotics, chloramphenicol, aminoglycosides, you're going to further inhibit mitochondrial function and increase your penetrance. But one of the most shocking things that Valerio Corelli found is if you look at mitochondrial DNA copy number, which is, by the way, variable in your cells, this is the number of mitochondrial DNAs relative to nuclear DNA for controls. These are affected individuals, and these are the unaffected individuals that are homoplasmic for the mutant. So what does that mean? That means that the genetic defect was compensated by simply making more mitochondria. So now we have an understanding for why you could have a homoplasmic mutation but not go blind, because there's a compensatory mechanism that increases your energy output to compensate for the defect. Okay? So that's a really, really interesting observation. And now we know that both the genetic background, haplogroup J, but also certain nuclear genetic variants are also affecting the penetrance of this disease. So what is J? Well, this is just a diagram of the migration of mitochondrial DNAs from the origin in Africa about 200,000 years ago in the Khoisan. Two different pygmies, sub-Saharan Africans and L3. These are L1, 2, 3. Of the sub-Saharan Africans in Ethiopia, we found that two lineages arose, M and N. They left Africa. M stayed in the tropics all the way to Australia. N went into the temperate zone to fill up temperate zone of Europe and Asia. And these are some of the European lineages that we classically talk about, HJ, TU, UKV, IWX, but there are also interesting lineages in Asia. And we believe that this geographic distribution of mitochondrial DNA is due to changes in the mitochondrial DNA that allowed people to adapt to different environments. But the point of that is that these European lineages then would be slightly different in biochemistry from the African lineages. And that's just shown here in a European lineage of the J haplogroup, which started with J with these two missense mutations, and then these two missense mutations that affect cytochrome B gene. And this affects an amino acid that's changed in all mesozoans. This affects an amino acid that's conserved all the way to E. coli, but these are polymorphic in this room right now. So these give you subtle changes in energetics, and they then augment the effect of the Leber's milder mutations. So now we can look at this mutation 3394. Remember, that was another one of the mutations in the mitochondrial DNA that increased the penetrance. It's an ND1 gene. So we can take blood platelets from individuals with these different mitochondrial DNA lineage mutations, fuse them to a cell without its own mitochondrial DNA, select for this nucleus but for the mitochondria, and take the cybrids, and then assay for respiratory complex 1. So this is a very severe mutation in the mitochondrial DNA, and you can see that it has a severe effect on complex 1. But now let's look at these lineages. These are N lineage, and this is M lineage. The N lineage B4, you can either have the 3394T or the 3394C, and you can see you get then about a 20% reduction in complex 1. Or if you look at the F lineage, 3394T versus C, here you have about a 15% reduction. If you're on the M lineage, then in fact this lacks some of the other mitochondrial variants that are in the N lineage, and then you see that even with the 3394C, you still have a pretty good complex 1 defect. So what is this telling you? First of all, yes, 3394C does give you a partial defect in complex 1, but look at the difference between F 3394T and B 3394T, or in fact M 3394C. So what that shows you is that you can't use a single polymorphic SNP as your indicator, because the entire mitochondrial DNA is acting together to give you the biochemical phenotype. So you have to look at what we call the haplogroup, the whole lineage together, all the integrated variants. So this is a study that we did with Chris Kenny and Nitkin Udar and all of their associates with Tony Newburn's patients population, and we just looked at these mitochondrial lineages, haplogroups, and asked what's the risk of adult onset macular degeneration. And what you can see is that lineages J, T, and U all have a substantial increase in risk for this disease. So here is now a background bioenergetic defect, none of which in and of itself is pathologic, but is increasing the risk of this disease. So this is then family where we have a mutation in the ND6 gene at proline 25 to leucine. This person had 50% mutant in her blood, and she had optic atrophy and cerebellar ataxia. This woman had 5% mutant in her blood and was perfectly normal, but with 3 different consorts, all of her children were 100% mutant, and they all died. So this just shows the kind of phenotypes we see in our clinic, where you'll have this woman coming in and saying, "Oh, my kids died," and then the internist has said, "Yeah, but you're perfectly normal. This can't be genetic," when in fact we're looking at segregation of heteroplasmy. So we can make a mouse. We can take cells and mutagenize the mitochondrial DNA, isolate the exact same mutation, put the mitochondrial DNA into embryonic stem cell, female, put it into a mouse, and transmit the mutant into the mouse, and get a mouse model of Leber's hereditary optic neuropathy. So this is the optic nerve of the normal mouse. This is of the mutant mouse. What's really interesting is when we look at synaptosomes, the defect only affects respiration about 30%, just like the 11778 and 3460. If we increase the energy demand, it goes up to near normal. But what happens is it doesn't affect ATP. It causes a huge increase in oxidative stress. So we really think that this is an oxidative stress primarily as the phenotype. And this causes alterations in electrocephalogram, gives you occult myoclonus, and also changes in the oscillation of the cortical electrical signals. So now we can take these mice and we can use a PET probe. This is a form of ethidium bromide, which when gets oxidized, it becomes positively charged. So when you put it into the animal, it diffuses throughout all the tissues, but when it sees superoxide anion, gets oxidized and gets fixed in that tissue. And then we put F18 on it as a PET probe, and we can actually then look at the oxidative stress in the different tissues. So this is the normal mouse, and you can see how oxidative the retina is. If we give this mouse a little bit of like LPS, which of course goes through the toll receptors, then we see that the optic disc lights up strongly, that now the whole cortex begins to light up, and if this is the whole body, you can see that the organs begin to light. But this is the mutant. Now you can see that the mutant without LPS is very similar to LPS, but if you give them even a subtle increase in inflammation, there is a massive inflammatory response. And we really think that that's probably the kind of trigger that drives a person from unclinical expression of Leber's into sudden onset optic atrophy. Okay, so now what's interesting about that is there's an inflammatory component, obviously. So why would that be? Why would energetics affect inflammation? Well, it turns out your T effector cells, which cause inflammation, actually are fairly glycolytic. This is oxygen consumption rate per cell number, and you can see by this seahorse method, they have pretty low oxygen consumption. But if you look at the T effector cells, T regulatory cells, they are much more oxidative. In this animal, as the animal ages, the T regulatory cells die off. And so what that's doing, since the T regulatory cells inhibit the T effector cells, then the T effector cells are becoming more dominant, and that's why then when we challenge them with LPS, we have a much higher inflammation response. And that then completely alters how the metabolism of the cells work. The T effector cells are going through glucose to pyruvate to lactate, whereas the T effector cells using FOXP3 can take lactate to pyruvate to oxidative phosphorylation, so they can then generate energy in a different environment. So then how is this related to anything that beyond a few rare 1 in 10,000 cases? Well, what's interesting, and this is work from my former postdoc in Trounce, if we now look at glaucoma versus the Leber's, and we look at complex 1 activity, we see the same kind of defect. And if we look at growth rates, the defect in glaucoma is very similar to the defect in Leber's. And if you look at the cells in the trabecular network, and you look at their oxidative stress, this is the normal and this is the mutant, and you can see that they have this massively increased oxidative stress. So we again think that this is the same pathophysiological mechanism, but this is just a milder form of Leber's. That's the working hypothesis. And we now have some haplogroup associations with glaucoma. So what are we going to do about it? Well, one of the most popular things to do right now is to try and use a genetic alteration to fix this disease. And this is a very messy slide, but it just shows that I did my literature search. But anyway, this basic idea is to take the gene from the mitochondrial DNA, change its genetic code to that's used in the nucleus, use a adeno-associated virus to put the gene into the nucleus, get the RNA expressed. The RNA is expressed, made into the protein. The protein then is vectorially transported back into the mitochondria, and hopefully you complement the defect. And there's a clinical trial both in France and in US on treating Leber's 11778 mutation by John Guy in US and my former postdoc Marcel Correll-Dobrinsky in France to use this allotopic protein import into the mitochondria to complement the 11778 defect. So far, we don't have any formal therapeutic benefit outcome data, although the indications are the toxicity is low. This is an idea from Jane Farah in Dublin using an old technique, and that is yeast has a single polypeptide for complex 1. It transfers the electrons but does not pump protons. So there the idea is you just put this protein in here and then stick it into the mitochondria and just bypass all the Leber's mutations. So far, none of us have gotten very far with that. And then the other one we're having a lot of fun with is you can take the mitochondrial DNA gene, keep its genetic code, but then put an RNA on the end that targets the RNA to go back through the mitochondria and get translated into the on the mitochondrial ribosomes. And it looks like that's now working, and that allows us then to actually put back in the normal messenger RNA and fix the defect. But those are all definitely in the works. What has been done is a clinical trial using idibinone, and this was a double-blind crossover trial. Idibinone is a CoQ analog, so it's a kind of antioxidant, and there was mild therapeutic benefit for Leber's patients. But what was particularly exciting to us when we were here many years ago with Dr. Levine, we discovered that in the mitochondria, the estrogen receptor beta is actually 20% is in the mitochondria matrix. And if we add estradiol, what we could show is we could double the manganese SOD, the antioxidant defenses. So the idea was then that maybe the reason women's were more resistant to optic atrophy is because they, in fact, had the estrogen and they were activating the manganese SOD. And if it's an oxidative stress disease, then they were reducing the oxidative stress by I don't even know what you call your own hormone, but by a classic hormonal therapy. So that was very exciting because then Valerio Corelli came up with a clever idea, I wish I'd thought of it, of using some phytoestrogens instead. And these are some phytoestrogens that are classically used in traditional Chinese medicine. And they happen to be specific for estrogen receptor beta and not alpha. And that's really important because if you do alpha, that feminizes. But if you use beta, you can use it for males and it won't affect their gender. So what he's able to show is that with these three classic traditional Chinese phytoestrogens, he could in fact regenerate the mitochondrial oxygen consumption of the severe Leber's mutations back up to normal. So now we have the possibility of using a traditional Chinese therapeutic that does not require going through large clinical trials and possibly even block the onset of this disease. So that's why right now we're working really, really hard to begin to survey different classic traditional therapeutic approaches to look for backbone molecules that we can then use to treat bioenergetics, with the idea that Western medicine is primarily about anatomy, Eastern medicine is about the vital force or qi, and the only thing that has both anatomy and qi in common is the mitochondria. So we think that that then brings us together with a new way of looking at these diseases and to understand them. And why is that important? Because one of the things we know is if you exercise, you'll increase by 30% your mitochondrial copy number. So clearly, all of the environmental things that Dr. Malik was talking about immediately target down on the mitochondria. And I'd just like to thank all of the really great people that did this great work. But since you've been so patient, I won't go through all their names right now. But thank you very much. So Dr. Zaragoza's thought is that let me digress. The advent of CRISPR-Cas technology as one technology, but there's also TALENs and a whole bunch of other approaches where you could actually use molecular biochemistry to change the genotype of a piece of DNA. So Carlos Moraes' laboratory in Miami has been using TALENs. These are polypeptides that when you get a pair of them that bind to a DNA, they then will cut the DNA at a particular sequence between them because of an enzyme. Restriction enzyme. He's able to put the TALEN genes in the nucleus of a cultured cell that would then see a heteroplasmic mitochondrial DNA mutation and go in and cleave only the mitochondrial DNAs with the mutation, thus eliminating the mutant mitochondrial DNA. And that's been, that is very well established now, and he's even proposed with another group that they might put TALENs into the oocytes of women that are heteroplasmic for a pathogenic mutation and actually kill off the mutant mitochondrial DNAs so that the normal could go on to development and give a normal fetus. But the other approach that there was one paper published out of Korea of possibly putting the CRISPR-Cas system in the mitochondrial DNA. And there you use a guide RNA and a protein called Cas9. Guide RNA finds the appropriate sequence, and then Cas9 will cut or alter that sequence. A number of groups, including ours, are working hard on that. There are some problems when you put Cas9 in the mitochondria, which you can do by targeting with a peptide. There's so much RNA in the mitochondrial DNA. The mitochondrial DNA is symmetrically transcribed, so all of there's double-stranded RNA for the whole thing. Then the Cas9 thinks that everything is target and just seems to degrade the whole genome. So we're trying to struggle with that problem right now. But I do think that certainly the TALEN approach is working very well. Certain restriction enzymes can be targeted to the mitochondria and remove known mutations. And so there's a growing interest in that approach. Another approach is to just physiologically alter the heteroplasmy level by physiological constraints. And so there's now a number of interests in using nutraceuticals to try and bias the energetics of the cell and make the wild-type mitochondria become more prevalent. Yeah. Yeah, so again, do patients with Leber's mutations have autoimmune disease? Do patients with mitochondrial mutations in general have autoimmune disease? I think that's an excellent question, and the reality is that this has not been rigorously tested. Then that relates to the problem we have in medicine in general. Most people will go to the specialist and will only be worked up for the most acute symptom. So whether that person will be followed over the next 5 to 10 years to find out if they subsequently develop some kind of autoimmune disease, that's not a part of standard of care. So I don't think we have good follow-up yet on our patients to really know that. But there is certainly good evidence that there is much more inflammation, whether that's autoimmune, where that's coming from. I think we need a lot more data on that. So I just think we just don't have the data to answer your question. So Dr. Cooperman gave an extremely important talk, and that is we can do a lot of observations, and we may or may not believe them. But until we do a rigorous clinical trial, double-blind crossover, we really don't know. So what do? I have the problem. When I started this business, I had people with complex multisystem disease, and they were all being sent to the psychiatrist because they thought they were crazy. We finally then showed they weren't crazy. They had mitochondrial disease, and they had mitochondrial dysfunction, and that's why they had all these symptoms that they were complaining about. My problem was fighting the mutation meant that I could say, well, before we were going to tell you we're going to die, now we can tell you why you're going to die. So we're in a very difficult situation that we want to do something. And the problem is that it's very difficult and very expensive to do the kind of elegant trial that Dr. Cooperman said. So the person on the front line, as you all know, you have to do something because that patient is there. So what people do is give mitochondrial cocktail. Coenzyme Q, we've looked really, really hard. It's very non-toxic. We've had a few cases anecdotally where it seemed to be miraculous, but we've had lots of cases where it didn't seem to do anything. Was this all fantasy? I have no idea. What do? I prescribe CoQ. I know it does no harm. Whether it does any good or not, I don't know. Do we use a cocktail? Vitamin C, vitamin E, beta-carotene? No. Zinc? Yes. There are multiple of these cocktails available, some of them commercial. Has anyone done a rigorous clinical trial on any known mitochondrial endpoint? No. Why is that? The reason is because mitochondrial clinical phenotypes are so variable. So let's say you have the most common mutation, 3243 mutation, that causes what we call MELAS syndrome. But actually, maternal inherited pedigree of MELAS only probably usually has one person with vascular strokes. By the way, vascular strokes might be a good reason that what you're seeing in your cases. But anyway, what you'll see in that pedigree is much more likely to have migraine headache. Migraine headache will track more in the maternal lineage than strokes does. Or if you do a muscle biopsy, you'll track ragged red fibers. Or if you look at people's ability to walk a certain distance, that'll track better on the maternal lineage than stroke-like episodes. But the only people that are going to come to the neurologist are people with strokes. So how do we do a clinical trial with vitamin E, vitamin C, zinc, and not beta-carotene on a family where every patient has a different clinical endpoint? There's no good way to do statistics. So why am I so enthusiastic about presenting to you labors? Because that's the one mitochondrial disease that has a very defined clinical endpoint. The only problem is that Nancy Newman and I tried to do some clinical trials 20 years ago with labors. The problem is we can only get 15 or 20 families. And so Nancy's idea, well, we wanted to get a person that had gone blind in one eye but not in the other, and then we were going to prevent the blindness. What do we get? 5 cases, so no statistics. So what we've done when my colleague, we can stand up here, my colleague Xiaogang Liu, we're developing an institute in Xi'an, China. Why will we do that? Besides, it's interesting. Well, my friend Minchen Guan in Zhejiang University has 1,000 labors pedigrees. Well, now I can do a clinical endpoint, okay? And so what our idea is that we can then use these classic traditional Asian therapies, but now have a big enough cohort that we know the people that are at risk for the disease, and we can look at their mitochondrial DNA copy number, we can find out whether they smoked or not, whether they drink, and what their mutation is, and we now can make pretty good prediction of what their risks are. And now we can use some classic therapeutic interventions and get some statistics. If we could then treat labors, maybe we could treat MELAS. But we'll never do a clinical trial in MELAS because we don't have an outcome variable.